two different excitation or emission wavelengths in order to quantify the results. But lifetime data is used at a single excitation or emission wavelength. Some reason that last thing that came, there we are. Lifetimes are required to quantify certain type of measurements. I mentioned anisotropy, polarization, quenching, fret, but to quantify and take those data and put numbers on them, you need to know the lifetime. And at a more fundamental level, lifetime determinations are required to understand the photophysics of fluorophore in diverse environments. Give an example, lifetime and quantum yields are dramatically affected by the environment in many cases. For examples that are relevant to this workshop are NADH. NADH in water has a lifetime of about 0.4 nanoseconds, but it varies, it gets longer bound to dehydrogenases. They can be as long as 9 nanoseconds in some cases. Other probes that are changed tremendously in the environment are ANS in water is about 100 picoseconds, but when bound to proteins, it can be 15 to 20 nanoseconds. Titanium bromide, 1.8 nanoseconds in water. Bound to DNA, it can be 22 nanoseconds. Bound to transfer RNA, 27 nanoseconds.
Mira qué lindo.
Oh,
Mira qué
I'm
lindo.
hearing somebody's needs to turn their microphone off. Lifetime of tryptophan in proteins changes a lot, from very short 0.1 nanosecond up to about 8 nanoseconds. So certainly, to understand a lot of these sort of studies, you need to be able to determine the lifetime. So, time resolved measurements are typically carried out in the vast majority of cases by either two methods, the time domain methods and the frequency domain method. And I'll be talking in a little bit of detail about both these methods because I think it's important for those getting into the field to understand how the data is obtained. But as I'll discuss in more detail later, phasors can be constructed using either time domain or frequency domain data. It's important to realize you still find people who believe, oh, phasors, you have to do frequency domain. I don't do frequency domain. No, it's not true. Either type of data you can do lifetime phasors on. Okay, so what is a time domain measurement? So typically you illuminate the sample with a short pulse of light, and then you look at the intensity of the emission versus time. Okay, so just a second. And here we see an example, a short pulse of light and then the decay of the intensity from some normalized value. Now, these short pulses originally, for many years, were done using flash lamps, which is a device, it had a width of several nanoseconds, and that made it very difficult to extract short lifetimes. But now we have laser sources routinely generate pulses with widths on the order of picoseconds or even shorter. So the idea is the intensity goes down with times as statistically some molecules emit earlier than others, okay? And when you get down to an intensity that's about 1 over E or 36.8% of the starting value, we call that time the lifetime. And I should note that typically you're doing lifetime measurements on populations, okay? But it's even possible now to do single molecule lifetimes. The idea with a single molecule lifetime, though, is you excite the sample that gives off light, and you do it again and again and again and again, thousands and thousands of times, and you build up statistically the same profile as you would if you had a population. So, as I just said, it's the time it takes to decay to 1 over E, and it's defined by this relationship. The intensity at any time is equal to a normalization or pre-exponential factor, alpha, times e to the minus t over tau, where tau is the lifetime. Now, it's been common for many years to plot the fluorescence decay on a log scale. And I have to say, the reason this started many, many years ago when I was a student too, and occasionally did some pulse lifetimes, we didn't have the computer to analyze quickly, and it was common to plot the log of the intensity, because then if it's a single exponential, you get a straight line as shown here. And it was an easy way to tell if you had single exponential decays. Nowadays, of course, you don't need to do this, but it's still is common to, if you see it, to show this this way of plotting the data. If you have more than one component, then you have a more complicated equation, which takes into account the pre-exponential and the lifetime values of the different components. And if we look at the log scale again, so if it was a single exponential, we'd get one straight line, but we might have, here we show two components well separated, and the short times you can see tau 1, and at longer times tau 2. Simply oversimplified, but it illustrates the point, okay? Now here's actual data of anthracene taken on a time correlated single photon counting instrument. I'll talk about that method in much more detail very soon. And we have a light emitting diode, very short pulses, the excitation. And here you see a log scale. You see the blue shows the pulse width of the exciting source, and then the data are the red points, and the green line going through it is the best fit, which in this case is 4.1 nanoseconds. And down below you see the residuals for the error in the measurement. And what one does is you try different models. In this case, single exponential is good, and we can see that the deviations of the given point to the best fit deviates above and below the line. And when you get an error surface such as this, you know it's a good fit because you're not deviating in one direction or the other. And this is called the chi-squared, which is a measure of how good the fit is. But let's look a little more detail. In the log scale, this is 36.8%. So this is the 1 over E value. And in this case, it took 73 channels from the excitation pulse to reach that point. So 73 channels, and it's 56 picosecond per channel. That's how you get to 4.1 nanoseconds. So that's the idea. Of course, you can collect fewer counts or more counts, but this is typically what one does. And with modern light sources, which have repetition rates of megahertz, it only takes a few seconds, very short time to collect sufficient data. Okay, what does the instrumentation look like? Well, we have a pulse light source, which sends a train of pulses into the sample. And then we collect the emission through a filter or a monochromator. And typically you have to use what's called neutral density filters to lower the intensity, because in this method, you're only allowed to count one emission photon at a time. But with the electronics, we first have something that monitors the exciting light. It can be a phototube or electronically. And ultimately, we go into this device called the time to amplitude converter. And that's the essential heart of these instruments. And what happens when we get the start pulse, let's suppose we have a pulse of exciting light, and we monitor when that exciting light arrives. That starts the TAC, that starts the TAC, and it starts a voltage ramp, which starts going up and up and up in voltage until the emission photon arrives. Now then, the voltage ramp, the voltage level that's reached, then addresses the computer, or we call it multi-channel analyzer, to put a point at that time. And this is illustrated here. So the first photon that arrives might go in this register. This next one goes in a different one, photon 3, a different one. And you repeat this process thousands of times until you think you have enough statistics, and you build up the histogram, basically, that I showed before, this one. So it's just a question of one photon at a time, And then, of course, if you're able to go at a very high repetition rate, you can get the histogram of the decay. So then you basically can get the lifetime from the slope of the curve, and you can fit it to different models, 1, 2 exponential, etc. Now, if the lifetime and excitation pulse width are comparable, you need the method called a deconvolution method to extract the lifetime. And here's an example of an old flash lamp type data where we have a very broad excitation pulse, and it's not trivial to extract it. In fact, huge effort over the years was expended on developing mathematical models to deconvolute or deconvolve the exciting pulse shape from the observed fluorescence. I remember when I was a student, well, right after I got my PhD, I went to a meeting in Scotland. Virtually the entire meeting was devoted on methods to deconvolve pulse shapes from the observed decay, which tells you how important it was. Nowadays, you don't even think about it, because for those of us in normal fluorescence with nanosecond lifetimes, the pulse width are so short we don't have to deconvolve anything. So that's a bit of history that took enormous amount of time and effort, and all of it is not useful unless you're one of the rare people that's looking at picosecond processes and excited state molecules, but that doesn't concern us. As Gregory Weber used to tell me, when you get less than nanoseconds, you're no longer looking at the public life of fluorophores, you're looking at the private life, and he thought it's best to leave them alone. So we will do that also. Now, what about the frequency domain? Well, in the frequency domain, the exciting light is not given in short pulses. It's usually a continuous source that can be, the intensity can be modulated sinusoidally at high frequency, like shown here. So the intensity of the exciting light will vary with time. And we used to always illuminate with a continuous wave laser, and then use a device such as a Paco cell, something to modulate the intensity. Nowadays, you have light emitting diodes or laser diodes that can be directly modulated. So we don't have to do that. And in such a case, the excitation frequency is described by this type of equation, where we have the excitation intensity at any time is equal to a starting level E0 times 1 plus ME, where M is the modulation factor. We'll talk more about that soon, sine omega t, where omega is the angular modulation frequency. And omega is 2 pi f, where f is the linear modulation frequency. So when we talk about something being modulated at like 80 megahertz, that's the linear modulation frequency. Okay, so this shows a little bit better, perhaps. Imagine the sinusoidal excitation, and it's characterized by a certain frequency of repetition, but also by what we call the modulation, the AC-DC ratio. So if we put DC at the center point here, then AC is how high we go above it or below it. Okay, and the modulation will be the ratio of the AC over the DC. Well, in such a case, the emission, because it stays in the excited state for some time, will be shifted in phase. And here we see it here by a phase angle, phi. Okay, and that's the phase shift, which will allow us to calculate the lifetime. But also the modulation of the emission changes too, because we're coming down and down and down, but then we get pumped up again by the exciting light. So we never reach the depth of modulation we did in the excitation case. So basically, we can measure the lifetime from the phase shift and by the change in this modulation factor. Okay, and the equation describing the fluorescence intensity is as follows, just like the excitation, but here we have, instead of sine omega t, we have sine omega t plus phi, where that's the phase shift due to the persistence of the excited state. So that allowed, actually in 1936, they derived the equation to do the lifetime based on the phase shift, and that was the tangent of the phase shift equal omega tau, where omega is the angular modulation frequency. And as I said, we can describe the modulation of the excitation and the emission, and the relative modulation of the emission is then AC/DC fluorescence divided by AC/DC of the excitation. And that allows you to determine a lifetime based on modulation, 1 over the square root of 1 plus omega tau squared. And that's a separate lifetime determination, separate from the phase shift, okay? And that became very important, especially in the early days of these measurements, because if you have a single exponential decay, then tau phase will equal tau modulation at all light modulation frequencies. But if the fluorescence decay is multi-exponential, then tau phase would be less than tau modulation. And that's how we used to judge in the early days when we had a couple modulation frequencies. Just how did the phase look relative to the modulation lifetime? And it changes with frequencies. As we go to higher and higher frequencies, tau phase gets shorter. And to give you a feeling for that, here's a typical data set one might get. And I'll tell you what's in this sample in a moment. But let's say I have an instrument. Now, this type of data is based on what we say cuvette measurements, not in the microscope, but it's the same principle. So here I illuminate my sample with 5 megahertz and I see a tau phase of 6.76 and a modulation lifetime of 10.24. So immediately I see it's not a single exponential decay. Moreover, as I increase the light modulation frequency, my phase lifetime gets shorter and the modulation lifetime gets shorter. And basically, it's those differences as a function of frequencies that allows me to fit the data to the underlying components, okay? So in this case, just shown, in the lab, I mixed 2 fluorophores. One was 12 nanoseconds and one was 1.4 nanoseconds. And I mixed them so that the relative contributions to the total intensity was close to 50-50, okay? Now, here I need to distinguish a very important point, okay, which over the years has caused a lot of confusion to many people, including me, okay? Namely, when we do this, we're doing this frequency domain measurement, we talk about the fractional contribution to the total intensity, and I usually refer to that with F. And that's different from the pre-exponential factor I talked about earlier. Specifically, you have to relate the terms. The fraction of the I component is equal to pre-exponential of I times lifetime I over the sum of all the pre-exponentials times their lifetime components. And in the case I just gave, the ratio between the lifetime is about 1/8. And what this means is there's actually 8 times as many molecules in that solution with a 1.38 nanosecond lifetimes as there was with 12.08 nanosecond lifetimes. Okay. Now, when it turns out that people, especially in the early days, people doing time domain would always report pre-exponential, and those of us doing frequency would always present fractional contribution. And for me, it made more sense, the fractional contribution, because that's the raw data. You know, you're getting how many fractions, what fractions of the photons you look at have this lifetime, what fractions have a different lifetimes. But it did cause some confusion. Nowadays, regardless of how you do the measurement, you can actually report, in most instruments, report both the fractional intensity and the pre-exponential factors. Okay, so that's something to just keep in mind if you ever look at the literature. Now, this is a typical plot for presenting frequency domain data. Here's a natural plot of fluorescein in 7.4 buffer done on a lifetime machine with the cuvette. And you can see these points are the phase shift as a function of light modulation frequency. And the red squares are the modulation ratio as a function of frequency. And typically you do multiple frequencies, and then this curve in the frequency domain is a single exponential decay. And believe me, after you've done enough of these, you begin to get a feeling just from the shape of the curve what's going on. And then I won't go into the details of fitting the data, but you get the best fit through the phase data points and the modulation data points. You don't fit the lifetimes, you fit the data points, okay? And in this case, these solid lines represent the best fit, which in this case was single exponential decay of 4 nanoseconds. Now, what if you have different values? This shows the phase in circles and the modulation in squares for ethidium bromide. Here we see 1.74 nanoseconds free in solution. When it's bound to transfer RNA, you get this 27 nanoseconds. And I know it's hard to tell from this figure, but these curves are the same shape. If you were to take the 27 nanoseconds and move it over to the right to higher frequency, it would superimpose on the 1.74. The only thing changes is the frequency response, okay? So this is what the single exponential decay looked like in the frequency domain, and it just moves back and forth along lower or higher light modulation frequencies. What if you have more than one component? Here in the black, we see 4 nanoseconds, and then in the red and the green, we have a mixture of 50% 4 nanoseconds and a 50% fractional intensity 1 nanosecond. And you see the shape of the curve is different. You can tell just by looking at it, these are different shapes. And again, you fit the data to see what's the best fit, whether it's one component, two components, or more, or a different type of decay. Now, I should comment that you don't need to have sinusoidally modulated light. In fact, many years ago, well, we see a measurement done in 1984. This is why Enrico and I had called a synchrotron radiation source, where you have pulses, and of course, you have pulses with the lasers now, and you can use the harmonic content of the pulses. Okay, so here we see, I think this is on the storage ring. Well, it's either Wisconsin or Italy. I don't remember anymore, but probably Italy. That's right. But you can see we could do the Fourier transform, and the harmonic content is shown below here, and that generates all the frequencies, okay? And in fact, that's what you do if you do frequency domain with 2-photon excitation. You usually have a Tie-Saf laser, for example, 80 megahertz, but you can do the harmonic content. Of course, in that case, it's 80 megahertz, 160 megahertz, 240. You don't have all the frequencies you would get where you can easily do it in cuvettes. Although nowadays, I think Lena will tell you some of their instruments, you have different frequencies. And is one measurement better than the others? Well, no, not really. This was an interesting paper done in 2007 where a group sent out different standard samples to a lot of different laboratories around the world, some of who did frequency domain, some of who did time domain, and they just compared the result. And as you can see, they're pretty consistent, you know, a little bit of difference, but basically both frequency domain and time domain got essentially the same result. So nowadays, in fact, usually don't distinguish. Like if you go to Enrico's lab, some of his instruments use time domain, some frequency, but as the operator sitting at the console, you don't know the details of how they did the measurement because you don't care anymore. You're just using the data. But it used to be a huge area of controversy and development as each of these methods improved over the years. Now, later in the workshop, well, you're going to hear a lot about fluorescent lifetime imaging, or FLIM. And because in FLIM, you get the lifetime data through the microscope, and you have lifetime data at each pixel in the image, and you hear about all these measurements. Here, see some example, like even though we see intensities of GFPs with different intensities at different parts of the cell, but the lifetime is homogeneous, you know, because it doesn't depend on the concentration unless you have some weird thing like fret happening. And as we said, it's more robust than intensity. And that gives it the great advantage, as you're going to see in many examples in this workshop. And just to let you know, it's not a brand new thing. Already in 1959, Veneta did lifetime measurements through a microscope, okay, using a frequency method. But that's pretty interesting to look at his work in 1959. So we have, well, more than 60 years of microscope lifetime measurements. Now let's talk about phasors. And this is an important part of the course. And now the concept of phasors actually has been around a long time. The original phasor transform was an invention of Charles Proteus Steinmetz, who was an outstanding mathematician engineer born in 1865. And in fact, around the turn of the century, he was a very famous figure in science and technology, made really significant contributions to electrical engineering. And he knew all the people of the time. And we could have a whole lecture on Steinmetz, but obviously we don't have time. But he introduced the concept of what he called phase vectors. That's where the word phasor comes from phase vectors, in a paper he published in 1893. And specifically, his phasor transforms really facilitated the analysis of RLC, resistance, inductance, capacitance circuits, to allow one to use algebraic equations in phasor space instead of differential equations in the time domain. Okay, so it was really an important development at that time. And in fact, if you search the literature, PubMed for phasors, some of the phasors you pull up are electrical engineering phasors. So you have to be careful when you're looking for these. But the inspiration for phasors and fluorescence was from a paper published by Gregorio Weber. And I want to mention this, and you'll hear much more about Weber in the history lecture at the end of the course. But at the time, when I was in Weber's lab as a student and then later on as a postdoc, we only had a few frequencies available, light modulation frequencies, but it wasn't trivial to fit the data, to fit the underlying components to the phase and modulation lifetimes. And Weber did this magnificent paper I show here, where he derived mathematically a closed form procedure to relate that if you have two light modulation frequencies and you have phase and modulation, you can analytically extract the two components. And if you had three frequencies, theoretically, you can get three components. But without going into the details of the paper, but it was really a mathematical tour de force. He derived in these papers, he described these terms called the G and the S, which are basically the equations we use for phasors today. And he did it for both frequency domain and time domain. So it was really a magnificent paper that he had here. Now, in 1984, Enrico and I, with Bob Hall, another student at the time, did a review on measurement analysis by using multi-frequency phase and modulation, because Enrico had recently developed the true multi-frequency, where you didn't have one or two, but variable frequency, as many as you want, light modulation frequencies. And in the appendix, we used a phasor diagram to discuss the application of this to some systems. And this is the modern lifetime phasor diagram. What was the first use in the lab? Well, the first use we actually put it to, one of the first uses, was to do online background subtraction. Now, let me put this in context. There was a paper with Greg Reinhardt, Pasquina, Marzola, myself, and Enrico. And the idea was this: back in the early 80s, and back in the 80s and the 90s, there was a lot of controversy between what's better, time domain or frequency domain. And one of the arguments the time domain people used was it was very easy in the time domain to subtract contributions from the background, from random fluorescence in the background, buffer or whatever. And they said you can't do that in the frequency domain. Well, with phasors, you can do that, because you basically did the phasor of the background and the phasor of your sample, and using the vector properties, you can extract the sample thing. And here we see we have this N and B that was NATA, N-acetyltryptophanamide, in a mixture with a lot of background. And this is the background frequency domain data. And here you can correct it to get the correct lifetime for any compound in the presence of the background. So that was the first use of that, actually, which then again was one less argument against frequency domain. Now, it turns out, I more recently discovered in 1993, Lenny Brandt's lab, a very famous fluorescence person, actually applied what now we would call phasors to look at multi-component systems. Specifically, he looked at Quin2, one of these calcium probes, which at lifetime and intensity change when bound to calcium. And he basically was able to study the binding of the calcium to the probe using what we now would call a phasor diagram. But they had not seen the 1984 paper nor the 1991 paper, so their work was totally independent, but they never named the plot. Okay. So let's talk about what lifetime phasors look like, and you'll see the analogous spectral phasor later. I think Lena was talking about that. So basically, when you do a phasor plot, you have this, what's called the universal circle. And as you have a point that, like, it depends on the phase angle, the longer the lifetime at any one given modulation frequency, the phase angle will get bigger and bigger, and it'll move along the universal circle. So basically, any single exponential decay is on the universal circle, and that's one of the major points. If it's not single exponential, it will be inside, or, and I'll discuss this later, if you have some excited state reactions, it can be outside. But the important thing for us, in the majority of cases we're looking at, the lifetimes will be on the universal circle for single exponential decays. For example, let's consider a lifetime tau 1. And the shorter the lifetime, the more it will fall on the circle lower here towards the 0, 1 ordinate point. And if we have another lifetime, a longer component, single exponential, it would be along here. Now, the whole key to the use of analytical use of phasors is that if you have a mixture of those two components, it'll be inside the universal circle, but moreover, it'll be along the line joining those points. And that's a very important point, this additivity property. Moreover, the fractional intensities can be determined by where along the line it falls. So it's immediately important to see that it's not exponential, but also, as we'll see, the big power of phasors for us now is that you don't need to assign to analyze the components, make this mixture. You want to see what happens to the mixture as you make some changes, some effects, okay? And we'll illustrate that. And here's a nice thing I borrowed from Lorenzo, who gave the nice talk the other day. Let's look on the left first. We're going to see two exponential components. We start with a blue one, and then it starts going down as the average component, the average profile, as we add a short one in. So we can see at the beginning, we have a long component that goes down, and the short one builds up to make the solid line the component, the composite mixture. Now look over here. This is what happens in the phasor transform. You start off with all of it on the single exponential B. You start adding another component, a longer one, and you see it gets moves along, the phasor point moves along the line joining them. So you can get a feeling how these things vary. And I think Lorenzo or other people will show you more animated plots like this. Now, the phasor approach, so it's been around since 1984, but it was pretty dormant until the last couple of decades. Several labs began to apply it to FLIM, but with different names. Quentin Hanley Andrew Clayton, Quentin Hanley, and Peter Vermeer made what they call the AB plots, Bob Clegg polar plots, and Enrico phasor plots. So let's look at the original paper and what Clayton and Hanley and Vermeer did. They wanted to resolve lifetime components in a FLIM image, and you'll see this is quite different from modern use of phasors. So they used Weber's algorithm, the one I pointed out in his 1981 paper, to extract. They made this plot they called the AB plot, which maybe wasn't the most inspired name, to be able to extract one component if you at one light modulation frequency. Okay, and they didn't really use present FLIM images. But Bob Clegg in 2005 described what he called a polar plot. And this is Bob Clegg in his lab at the University of Illinois. And he unfortunately died some years ago, but he was a magnificent scientist in person. But he had some beautiful papers on this, what he called a polar plot. And they didn't present any FLIM images, but they wrote actually in this paper that they've been applying the methods for several years to FLIM data. But both of these references were to abstracts at Biophysical Society. So we never got to see what FLIM images they actually utilized. But Enrico introduced phasors for the first time in his lecture at the 2005 Principles of Fluorescence course in Genoa. There's Enrico there. Here I am. And then in 2006, he started to give his famous LFD workshops, and he defined this more clearly, where he gave lectures on applications of phasors to FRET analysis. But these lectures delineated the modern use of phasors in FLIM. And I want to emphasize that this method is conceptually completely different from previous work on FLIM and phasor-related plots. Specifically, he introduced the way to connect the pixels in the FLIM image, so we can imagine that here, to the phasor points from these where each pixel would lie in the phasor diagram. And this becomes extremely important, as you'll see illustrated very well in this course. And this graphical approach became a transformative tool, and now there's hundreds and hundreds and hundreds of papers published using this phasor approach to FLIM. Now, over the years, Enrico always invited me to these LFD advanced workshops. Presumably it was for comic relief, but that's another story. And it was during the 2008 workshop, I was listening to Enrico expound on the wonderful merits of phasors, which I admit I had not fully appreciated until then, when it occurred to me, wait a minute, I could be doing this in a cuvette. There I was post-tenure in Hawaii. So that happy thought kicked out a series of cuvette-based phasor measurements from my laboratory. So interestingly, we first turned the paper into analytical chemistry, where the reviewers rejected it because they said they didn't think the phasor method was of any value whatsoever. Fortunately for us, it was accepted to analytical biochemistry. We have two papers with Nick and Justin, my postdocs, and Martin Steffel, a grad student from the Czech Republic. And we even made the cover of that issue. But I think it illustrates in these papers the principles of phasors. So here we see actual data in the laboratory where at 20 megahertz light modulation frequency, and again, you can use any frequency you want in the cuvette measurements, we see three single exponential components: DNS about 30 nanoseconds, AEDANS 5.3, and rhodamine 1.7. And they're all single exponential, and they lie along here. And now you start mixing them, and you can see if you mix DNS and AEDANS, all the points lie along this line joining them. DNS and rhodamine along this line, and AEDANS rhodamine along here. Then you can make the ternary mixtures, and they lie along this line and going to one particular mixture here. So again, you can see how it lets you see that there's components inside, and you can actually analyze those to get knowing the fractions you can solve for different things. And I'll illustrate that. Then here's what happens when you do two light modulation frequencies. Normally, in most of the phasor plots, you'll see And from the FLIM images, you tend to use one light modulation frequency, but you don't have to. In this case, I just want to illustrate what happens when you change the light modulation frequencies. It basically rotates those images around. And the point of this is you can basically, to illustrate a point, which I'll do several times now, you can pick the frequency that best illustrates your point. Now, one of the early things we did was look at tryptophan, and this figure is from my thesis, just looking at the phase lifetime and intensity of tryptophan as a function of pH. And when you protonate and deprotonate that amine group, it dramatically changes the lifetime. You go from 3 nanoseconds up to almost 9 nanoseconds. Well, of course, that gives the idea that you can do a phasor plot, and basically at pH 11, you're over here at like 8.5, 8.7 nanoseconds, I think. At pH 6, you're down at 3. But any pH in between, you'll follow along this line in a known fashion. You just have to use the famous Henderson-Hasselbalch equation to get the fractions if you want. And you change the light modulation frequency, it just moves along that. But the point is, again, you can see what's happening, that at the two extremes, you have single exponential, and in between, you have a mixture. Here's another illustration using an N-acetyltryptophanamide, a famous tryptophan analog. 3 nanoseconds, here we see the phasor plot at 89 megahertz, and then you start quenching it with acrylamide, and it quenches, it remains a single exponential as you go along the universal circle. You see it getting shorter and shorter. What if you have a protein with multiple ones? Well, here's lysozyme, which has 6 tryptophans. Most of the fluorescence comes from 2 of the residues. And you see these points inside the universal circle, and you see how it moves when you quench it. Well, we're going to see how you can use this to quantify processes. And one of the early things I did too, I work on with dynamin, a GTP binding protein. It has 5 tryptophans, but again, it doesn't matter how many tryptophans you have, you'll get one point in the phasor plot, and you add GDP, that moves the lifetime. So you're affecting the lifetimes of one or more of the tryptophans. You add GTP, non-hydrolyzable analog, it moves differently. Here we have monomeric HSA. HSA has a single tryptophan. It's almost single exponential, but not quite. But if you get HSA from a bottle of lyophilized HSA, many people don't realize you have about 15% of a dimer. You get cross-linking of sulfhydrose, and that lifetime is different. And I illustrated that to show that people that study HSA right from the bottle, it's a difference. You have 15% of quite a different lifetime. We isolated the pure dimer from a column to do this. You add thyroxine, which binds, it changes it. Furosemide, it changes it. So you can immediately study properties of tryptophans and proteins. And this is a study where we show we have 2 proteins, antithrombin and lysozyme. They don't interact. Each of them has multiple tryptophans, but you put a mixture of them together, add one to the other, and you get a data point in between. It's on the line because it's a mixture of non-interacting species. But suppose they interact. Here we have thrombin with multiple tryptophans introduced into antithrombin. They bind, and the phasor point moves off the line. So this is a way to tell that 2 proteins are interacting without without labeling the proteins or anything. So, and that's something we can use now in some examples. But first, let's look at denaturation. We all know that many protein aficionados like to put denaturants like guanidine hydrochloride or urea in their sample. And we can see when you add it, these phasor plots are different. And this was shown very beautifully by Felipe Montesinos, a Chilean student who came to my lab some years ago. And they were working on a protein called PHITC, which is a bacterial protein involved in splitting of the cells, and it has no tryptophan. And here's the data when he put in a tryptophan in position 40. And you can see he made very detailed titrations with urea. You can really see the phasor plots move in a reproducible manner. Then he went on to look at 3 different single tryptophan versions of this protein. And now this is a 3-dimensional plot, but the actual data points are shown here. This back here on the left is the projection along 2-dimensional axes, but it's beautiful to see in 3 dimensions. And you see they start off quite different, but as you denature with urea or guanidine, they all come together. So it gives you really detailed pathways of these unfolding processes depending on the denaturing, or you do it with temperature or other variables. Now let's go back to this, one of the papers by Bob Clegg, who did a beautiful paper looking at what happens in fret. And here we see a donor. This is all simulated, 3 nanoseconds. And as you quench it due to energy transfer, if it starts off single exponential, it will retain, it'll still be single exponential, but shorter and shorter as you quench it more and more. But interestingly, he also showed what happens when you look at the acceptor fluorescence. So we're exciting the donor, but we're looking at the emission from the acceptor, and we see it's outside the universal circle. And this is the important concept, because we excite the donor, and there's a certain phase delay in that process, but then there's a phase delay before we transfer to the acceptor. So we're adding an additional phase into the process, which takes it outside the universal circle. And we'll see that both for fret and for, with Lordan, we'll see relaxation processes. Now, one of the first things we did was test this in, I think, an interesting case. We looked at the chromophore in green fluorescent protein. And I think, as most of you know, that's formed in the interior of the protein in this beta barrel structure by 3 amino acids, glycine, serine, and threonine, 66, 67, 68, I think, coming together to form the chromophore. Now, regular normal GFP has 1 tryptophan residue. So if I were to ask you, is there energy transfer from this tryptophan to that chromophore, how would you answer that question? In many cases, to say that, you would look at the donor in the absence of the acceptor. Well, you can't do that here. So what we did with phasors is you look at the phasor point for the chromophore, and you're exciting first at 471. You're directly exciting the fluorescent chromophore, and you're looking at emission greater than 500 nanometers. And we see all the points around the universal circle, regardless of the light modulation frequency. Now excite that same chromophore at 280. So you're exciting it at 280, but you're looking at the emission of the chromophore greater than 500. All the points are outside the universal circle. This immediately tells you there was energy transfer. I'm not trying to resolve the component magnitudes or anything, but I'm looking to see, yes, there was energy transfer, okay? So immediately it illustrates how useful these phasors can be in such a case. Now, let's talk about Lordan. You'll hear much more about this later, so I'm not going to say a lot about it. But Lordan can go from we call the initial, or the initial state to a relaxed state if dipoles can relax around it. And this is a relaxation process, and that also will cause a shift, a phase delay in the lifetime that takes it outside the universal circle. And here's some data that Leonel took in my lab in Hawaii. And let's look more closely. He's looking at mixtures of DPPC and then with different amounts of cholesterol, and as a function of temperature. And you can see the points in some cases are completely outside the universal circle. And again, they're reproducible, but there's no way to describe this data using combinations of exponential decays. But you do get a fingerprint of what's happening and get information, again, from the phasor plots. Here's an example of Jose Lopez, from a postdoc in my lab, came from Brazil, and he's working on peptides derived from an enzyme, diacylglycerol acetyltransferase, and his peptides had a couple of 1 or more tryptophans in them, and they bind to micelles. So here we see free and bound, and we see that in the absence of my cells, the point came here for those two tryptophanes in the peptide. Now you put in an excess of my cells and it goes to a different location. Then he did the careful titration down here. And here's that region he titrated along the line. You can see it's a beautiful straight line. So you say, how do you analyze this and change this data to a binding isotherm? And we had the happy thought, this is finally the occasion to use the Pythagorean theorem that we all learned in high school, right? Because it's the distance along this line, and we know the square of the hypotenuse is the sum of the squares of the other two sides. So that's the sum of the squares of the other two sides plotted against lipid concentration. You get a beautiful binding isotherm. Again, not labeling the protein or doing any other method, but just look at the phase data. The final example I did actually in Uruguay with students from and postdocs from Anna Danocola's lab, and their biological problem was they had a protein peroxyredoxin, which was a decamer, so 10 things, and it dissociates to a dimer, they thought, from column work, etc. But how do you follow this in solution? And we tried different things, labeling it with fluorescent probes, doing polarization. But then it occurred to us there's two tryptophan residues, and you could do a standard lifetime analysis and get different data, but you don't have to do that for this experiment. Now, I should say that normally to study a protein's tryptophan lifetime, you excite at 300 nanometer, which avoids tyrosine to tryptophan transfer. We don't care about that here. But there's much less absorption at 300. But at 280, you don't care if you excite tyrosines. You don't care how complex it is. You get one phasor point at any concentration. All you're hoping is that the lifetime changes when you have the decamer to dimer transition, which is not unreasonable. If the protein dissociates from a decamer to a dimer, probably one or more tryptophan will be affected. And it does happen. And here you see 40 micromolar, the phasor point. You dilute and dilute and dilute, you get to 5 micromolar, it's changed a lot. And in fact, what we're seeing here is the decamer to dimer dissociation. Now down here below this, when we originally did it, we thought this might have been a dimer monomer. Well, it's not. Turns out it's just you get weaker and weaker signal because you're diluting and you're picking up fluorescence in the buffer. And you'll see when you do the in vivo work with phasors, you'll see a lot of autofluorescence, the background. And when you clean this up, you don't no longer see that. And you see this beautiful dissociation curve. So you can dissociate it very quickly. It's just measure the phasors, dilute, measure the phasors, dilute. And you can do it at different temperatures. And we did a lot of work, and that's published now. So back to phlegm. So Enrico Gratone's phasor approach, and you'll hear all about this from the man himself. Enrico, next lecture, you get a phasor point for each pixel in the image, so you can go back and forth from the image to the phasor diagram. And you're going to hear several lectures, people using the lifetime phlegm microscopy and phasor plots to look at metabolic states. Okay, and this was the first paper. Now, these things here, it's not one image. They did, where's the phasor point in cells for free NADH, bound, FAD, other things. So it's a composite of where different things, autofluorescence components appear. Okay, and you're going to see this expanded upon in great detail and see how valuable these measurements are and how very, very popular they are now.